Protein kinase C PKC signaling pathway. Signaling molecule will go on to activate the G protein coupled receptor. G protein coupled receptor will go on to activate GQ. GQ will go on to activate PLC beta. PLC beta will go on to activate DAG and also IP3. IP3 will go on to activate IP3 receptors. DAG will go on to activate PKC partially. IP3 receptors will go on to activate calcium, increasing cytosolic calcium concentrations. Calcium will go on to activate ranitidine receptors. Ranitidine receptors will go on to increase even further the cytosolic calcium concentrations, thus activating calcium even further. As calcium concentrations increase, there's a negative feedback loop both on the ranitidine receptors and the IP3 receptors. And as we said, DAG goes on to activate PKC or localize it to the membrane. And with the effects of calcium, you have full activation of PKC. And this is being shown in this figure here. Now we're going to look at the first initial part of the pathway. So the G protein coupled receptor and GQ, the G protein, in their inactive state, where the G alpha and the beta gamma subunit are bound to each other and the G alpha subunit has a GDP. The G protein, trimeric G protein of the PKC pathway is called GQ. Upon binding the signaling molecule, the G protein coupled receptor is activated and the G protein, GQ, binds to the G protein coupled receptor. The G protein coupled receptor acts as a guanine exchange factor, which will increase the rate by which the GDP of the G alpha subunit is exchanged for GTP. Upon the G alpha subunit binding GTP, the G alpha and the G beta gamma will dissociate from each other, and of course, they're both dissociated from the G protein coupled receptor at this point. Upon dissociation, both the G alpha subunit with its GTP bound and the G beta gamma are in the active state. Both of these subunits, the G alpha and the G beta gamma subunits, will diffuse laterally on the cell membrane. Now we're going to look at the second part. The G beta gamma subunit is the one that is responsible for activating the PKC pathway. The G beta gamma subunit will go on to bind to PLC beta, and this will lead to the activation of PLC beta. What does PLC beta, phospholipase C beta, do? PLC beta is an enzyme that catalyzes the following reaction. You have PI45P2 is a phospholipid that has been phosphorylated twice on its N-acetyl head group. This is also called PIP2. So here's an N-acetyl head. Here are the extra phosphates. And PLC beta will catalyze a cleavage reaction, a hydrolysis reaction at this point being shown by the arrow. And the product of the hydrolysis reaction will be diacylglycerol, DAG, and N-acetyl 1,4,5-triphosphate, or IP3. These are both second messengers. Now we're going to look at another part of the pathway. PKC will bind to DAG, or it can be said that DAG localizes PKC to the cell membrane. This is a partial activation step as calcium is needed for full activation of PKC. And this is being shown in the figure that we had initially, here where there's that red box. Now we're going to look at the last little part of this pathway. IP3 receptors are ligand-gated calcium channels on the ER membrane. The ligand for IP3 receptors is IP3. As IP3 concentrations increase due to the actions of PLC beta, IP3 receptors will open. Calcium will flow down its gradient from the ER into the cytosol through the open IP3 receptors. Calcium concentration in the cytosol increases. The increase in calcium concentration in the cytosol will lead to the opening of ranitidine receptors. Ranitidine receptors are calcium channels on the ER membrane that open when cytosolic calcium concentrations increase. This is a positive feedback loop, being shown here. At low to medium cytosolic calcium concentrations, ranitidine receptors are opened. High cytosolic calcium concentrations will inhibit both the ranitidine and IP3 receptors by inactivating them until cytosolic calcium concentrations decrease. This is a negative feedback loop which leads to a calcium wave in the cytosol of the cell when IP3 is released. And we have the picture, including now both the positive and negative feedback loops. And this picture that is in the study guide is showing you the calcium wave across the ER membrane on the cytosolic side. And again, this is a picture from the study guide that I went over in a previous video. We know that PKC was localized to the membrane by DAG, and now, with the help of calcium, will go on to be fully activated. Once fully activated, it will go on to activate cell-specific effector proteins, which will lead to cell-specific responses. And in this table, you can see that different cell types in different tissue will have different responses upon the activation of the PKC pathway.